welcome guys to the next episode. Good to have you back, and if you're new, welcome on board. Okay, so we're continuing our discussion, and in year 2 we are talking genes. Right, we're talking genes. And last time we talked about mutation, and I guess the idea that's tying all this together is, what are the things that can affect the function of a gene? Okay, because genes make proteins, or genes give rise to proteins, and essentially it's proteins that carry out the function of the gene. Okay, and what we discussed last time is that, well, there are certain things that affect the functioning of a protein. So if you have a change to the sequence of bases in the DNA of the gene, if those sequence of bases are altered by a mutation, then that can change the function of a gene, because it's going to change the shape of the protein, and we don't need to go into all of that again, as I have a tendency to do, because this is in fact take 3, and I'm trying not to recap or rehash previous episodes all over again, so control myself. Okay, so mutation is one way that you can control or change the function of a gene, but that's kind of unregulated, right? That's unregulated. That's not really something that is desirable. Okay, yes, over millions of years it can lead to positive change, but on a daily basis you don't want to be mutating all the time. Okay, so that's unregulated, and generally speaking undesirable, right? Because you've got proteins that are doing a job. They're allowing the organism to function, the cells to function, and so we don't really want mutations. Okay, however, we do want to modify the function of genes. We want to modify the function of proteins in a controlled way. Okay, so in a controlled way, we want to be able to tweak the function of these genes. We don't just want them to be working all the time in every single cell. Okay, so being able to control the function of a gene is what we call gene regulation. Okay, so in a controlled way, we modify the functioning of the gene. For example, like if you go into your car, if you have a car, if you go into a car, right, and just imagine what it would be like if the only 2 speeds a car had was off, i.e. not moving, and 100 miles per hour. Okay, that's going to be detrimental for getting from A to B safely. Okay, so there's got to be more to the working of genes than simply go as fast as you can. And organisms are so complex, and they go through so many different circumstances, scenarios, and environmental conditions that they need to survive, that they kind of need to be able to respond to things as well. Okay, so it can't just be this on-off situation. So again, just because a gene is there doesn't mean we need its function all the time, and that means we need to be able to control it, and controlling the function of a gene is what we call gene regulation. Okay, and it's another way that a function of a gene can be manipulated, but again, to stress, this is controlled, regulated. This is what we want. Okay, why? So we're gonna answer a few questions. Okay, and again, in this episode we're gonna keep it short and sweet. I'm gonna introduce the concept of gene regulation. We're gonna talk about the different situations in which we might want to do that. And finally, we're just going to mention, without going into too much detail, we're just going to mention what are the ways in which gene regulation is carried out. Okay, and we will leave it for subsequent episodes to do deep dives into the different mechanisms of gene regulation. So let's get into it. So what are the questions? Well, one, what is gene regulation? And I guess we've already talked about that, right? We've already discussed that. Gene regulation is manipulating the function of a gene in a controlled way. In a controlled way, not a random way, okay, like mutation is, because mutation affects the function of a gene, but it's spontaneous, it's random, it can happen at any time, and in a way, and with unknown effects, okay, or so it's difficult to tell what the effect of that mutation is going to be, all the way from no effect all the way to protein not working properly. Okay, gene regulation is a really specific and controlled way of modifying gene function to achieve certain functional goals. Two, so the second question we're going to be talking about, then the next thing we're going to be moving on to is why? Why would we need to regulate genes? Why? Okay. And three, how? How do we regulate genes? Okay? Or how do we regulate gene function? And that's going to be the scope of this little episode right here. So let's move on to the second question. We've discussed what gene regulation is. And now we're going to move on to why would we want to regulate genes in the first place? Well, if you're an organism, there are two key things that you have to do with the genes that you have, okay? First, the organism has to form, okay? It has to form if it wants to survive. Actually, let's do this properly. If an organism wants to survive, this is the argument that I'm going to make here. An organism it exists in order to survive and reproduce. If it wants to survive, it needs to do two things. It needs to have its structural adaptations. It needs to have its structural adaptations, which means it needs to have its anatomical features, shall we say. Anatomical features. Okay? So if you're an animal, you need to have if you're an animal that moves around, you need to have legs to help you to move around, because having legs allows you to capture your prey. It allows you to perhaps evade your predators, meet with your mate. Okay, so in order to have the best chance of survival and reproduction, you need to have your anatomical features. You need to develop. Okay, and you need to have that differentiation. So I'm going to just chuck a few words here which all belong to the same idea, which is specialization. You need to have specialization. If you want to form an arm or a leg or an eye or a nervous system or whatever, you need to have specialization. You need to have differentiation. Okay, I hope this is making sense. I hope you're believing my story. It sounds logical. So in order to have that, and remember, all the cells in an organism have the same genes. All the cells in an organism have the same genes. So why is it that muscle cells are structurally and functionally different from neurons or nervous tissue? And why is nervous tissue structurally and functionally different from liver tissue? Right? Even though they have all the same genes. So we have to think about that. We need to think, how do we create cells and tissues that are so distinct from each other even though they have the same genes in their nucleus? Right? And that is because we can regulate those genes. We can regulate those genes. We can say, right, if you're a muscle cell, you can switch the genes for actin and myosin on. Because as a muscle cell, you're going to need those proteins, right? Okay, and so, if you're like a stem cell or something, and you're going to differentiate into a muscle cell, if you're a stem cell and you're differentiating into a muscle cell, well, in order to do that, let's just zoom in there. Okay, in order to turn a stem cell into a muscle cell, we need to be able to switch on. So genes for actin, myosin need to be on. We need to express those genes in a muscle cell. But in order for this muscle cell to be a muscle cell, do we really need the genes for, I don't know, insulin? Or do we need the gene for collagen to be expressed inside these muscle cells? Perhaps not. So, or hemoglobin, right? So, while some genes need to be switched on for this muscle cell to become a muscle cell, other genes, we need to keep them switched off. We don't want them. We don't want the proteins to be produced from some genes because the muscle cell doesn't need those proteins to be a muscle cell. Hope that's making sense. So, in order for an organism, right, to be able to do that, we need to be able to turn genes on and we need to be able to turn genes off. Okay? But that's important in time and location. That's important in time and location. So, as an embryo becomes a fetus, becomes a baby, becomes a child, becomes an adult, in order to generate those changes in physiology, in structures, in order to get those changes to happen in particular places, so fingers form where fingers are supposed to be formed, eyes form where eyes are supposed to be formed, etc., we need to make sure that those genes are expressed in the right cells, which are in the right place. So those genes need to be not only on and off correctly, but they need to be in the right place. Okay? And similarly, as we just discussed, all these changes need to be, temporally happening in the, or temporally controlled as well as spatially controlled. So we need to express those genes at the right time to ensure that, the baby is not developing structures that are only supposed to be formed when, in adulthood. Okay, so genes, the first idea is genes can be on and genes can be off, and that's important. The second idea is that genes need to be expressed in the right places for the right structures to be formed, but not only in the right places, but at also the right times. Okay, and that's important. So you got structural adaptations, but we also have functional adaptations, or I might also include behavioral here. Okay, so and what I'm talking about here is being able to respond both to environmental signals and also to internal signals, homeostatic. Okay, and if you want to be able to respond, to the right signal. So if it's starting to get cold, if the environment's starting to get cold and the body temperature, it causes the body temperature to go down, well then the organism has to respond by doing certain things to increase its body temperature, right? If the blood glucose level is getting too low, well then the organism has to respond in some way to raise that blood glucose level so that the organism can continue to carry out respiration in all its cells. These are responses, and they're just as important to the survival of the organism as its anatomical features. Okay, now responses, responding to the environment, changing the behavior, this is all to do with changing the behavior of cells, right? It's all to do with that. So if you want to make a response, if you're an organism and you want to make a response, then that comes down making any kind of response, whether it's a response to an environmental signal or an internal signal or stimulus, it comes down to changing the behavior of cells. Changing the behavior of cells. It comes down to changing the behavior of cells. And how do we change the behavior of a cell? We can change, or we can modify the gene regulation, right? So for example, e.g., okay? Let's say the blood glucose goes up. Okay, well, we need to lower it. So what do? Well, we don't, but the pancreatic beta cells, okay? So the beta cells, pancreas, what can they do? They can detect that the level of glucose has gone up, and they produce insulin. So we need to, the gene in the pancreatic beta cells, the gene for insulin is switched on, right? So we produce insulin in response to the high glucose. So when the glucose level goes up, the beta cells in the pancreas, the gene for insulin is switched on, and so those pancreatic beta cells are producing more insulin. Okay, we didn't need those cells to be expressing or producing the insulin before, but we do need it now. Okay, and so we need to be able to turn this gene off, and then at a later point when we've controlled the blood glucose level, well, at that point we won't need insulin anymore, and so at that stage we can switch the gene for insulin off. But I hope you can see why it's important to be able to turn genes on, switch them on or switch them off as per the requirements of the body to make responses, but also, even the more fundamental things like developing normally, right? So that is why we need to regulate the gene. Okay, so how do we regulate genes? Let's just remind ourselves of how we go from gene to function, and bear with me, I might just introduce a few new details here. So we have a gene, sequence of bases in the DNA. Okay, now the first thing that happens to the gene is transcription. Okay, so I'm just going to write that process here, transcription, and it produces an RNA molecule, right? A section of the DNA unwinds, we make an RNA copy of the gene sequence. Okay, but then we don't get the mRNA straight away. We get something called pre-mRNA. Okay, and it is another process called mRNA or RNA processing. Okay, this is called RNA processing, again to be detailed later on. RNA processing, and RNA processing is what gives us the final mRNA that is going to exit the nucleus, bind the ribosome, and be the instruction to build the protein. Okay, so once the mRNA then exits the nucleus, it is going to bind the ribosome, and the mRNA is going to undergo the process of translation, right? Translation, again, not new. Okay, translation, and translation is going to give us the protein or polypeptide, which will eventually become the protein. And the protein can undergo modifications, right? We know about this because that's basically the whole job of the Golgi apparatus, is to modify proteins, and anything that modifies a protein's structure is going to affect its function. So there we go. So finally we have what we call post-translational modifications, right? So after the amino acids get put together, that's not usually the end of the story for the protein. Either it might need to be joined with its other subunits to form the quaternary structure, it might need to bind with a prosthetic group, it might need to bind some cofactors or something else before it becomes the final functional protein. But after all of that, then we have the functioning protein. Okay, now if you think about it, then look at the journey, look at the steps we have to go through, from all the way from the information in the gene all the way to the final function. Okay, so if you can intervene anywhere along this journey, you can affect the final function of the protein. If you can tweak any of these processes along the way, you can regulate this process to either increase the function of the final protein or reduce the function of the final protein. So what can you do? Just gonna note them down here. First, obviously, is transcription. You can do a number of things to affect how much transcription occurs. Because if you get more transcription, you're going to make more mRNA, which is going to lead to more protein. But if you reduce transcription, you're going to get less of the mRNA and you're going to get less of the protein, therefore less of the function. As and when and where you need it, as we've discussed. Okay, so that's the first one, transcription. 2 is RNA processing. Because even once you have copied the DNA into RNA, there are still things that are happening to the RNA before it becomes the full-fledged mRNA. So anything you can do to increase that process or reduce that process is going to affect the function as well. 3, we have things that affect how much of the mRNA actually gets translated by the ribosomes to make the protein. So obviously that's going to affect the function. And 4, we come back to those modifications that are made to the polypeptide or the protein before it becomes that final protein. So any of those post-translational modifications could affect the function too. Okay, and there we have that, guys, and that's where I'm going to leave it. So in the next episodes as they come, we'll discuss these different mechanisms of affecting the function of genes by gene regulation. Thank you for being here, guys. So because this is an ongoing series, if you give me some feedback on the videos, it can impact on how I'm doing things going forward. I can modify things as to what might be more useful for you guys. So that'd be useful if you leave some comments for me as to how the series is going. Also, if you have questions, I would love to be able to respond to questions in a more kind of real-time way. So if you have questions about any of the things that we've discussed in any of the episodes, in subsequent episodes I can address those issues. So that would be good to do as well. All right, guys, thank you for listening. I hope it's been useful to you. Until next time.